Here is Dr. Scott Walmsley.
Hi, thanks, Nick. So the title of my talk is Computational Support for Mass Spectrometry-based Omics Research. I am currently working with the Analytical Biochemistry Shared Resource and am under the direction of a couple of people, Rob Turisky at MCC and Xinhua Wang over at the Institute for Health Informatics. A little bit of a personal background where I come from. I did both my degrees at Colorado State University. It didn't take me 15 years to get a PhD, but I had a stint in industry, did my work in the area of mass spec-based proteomics and some developing informatics under Norman Kurthoys in the area of renal proteomics. I completely went computer at the University of Michigan for Alexei Nizhvisky in proteome informatics. After that, I ended up at the University of Colorado, Denver to learn a bit about metabolomics under Nicole Reisdorf. And now I'm lucky enough to be here at the Masonic Cancer Center working on DNA adduct omics for Rob Turisky and Xinhua Wang, and now developing into something which will hopefully be the greater community here at the MCC. So the outline is, I'll give you a bit of background, what do we mean by omics, what do we mean by LCMS, and then I'll dive into a couple of informatic projects that we have developed over the course of the last year and a half and are now being put into practice. Give you a little overview of some metabolomics data analysis that we have completed in the past, and mostly for some poster work and conference work, and then hopefully here again soon for some reproducibility studies. And then also a description of proposed services that we feel hopefully some of you might be needing in the near future. So the background: development of informatics services and the omics defined. So informatics at the Analytical Biochemistry Shared Resource. Why are we pushing forward this concept? The Analytical Biochemistry Shared Resource provides mass spectrometry capabilities for research needs. There's a lot of data being produced in the core facility there. Data that is produced also are increasingly producing larger and more complex data sets, particularly off mass spectrometry platforms. And there's increasing interest in DNA adductomics and metabolomics, and I will describe or define what those are. There have been other mass spectrometry-based needs for data analysis. So beyond DNA adductomics and metabolomics, there's been a number of maybe small, more discrete studies where some folks have needed some statistical help. And then also we want to maintain some better accessibility to software and expertise that may otherwise have been more difficult to have. And then we want to formalize our access to these informatics services and build it out from there. So the informatics relationship between the analytical core and the Institute for Health Informatics looks something like this. Peter Villalta is the director of the ABSR. So if you're doing a mass spectrometry assay, he's your point person over there. Rob Turisky brought me on to the Masonic Cancer Center, and I've been very excited for working here with him and his group, and with Pete, and of course with Xinhua Wang, where we kind of gained that academic computational expertise. And so really we see the informatics component serving as a bridge between the analytical and the computational components. So omics defined, genomics and transcriptomics, I'm pretty sure we've all heard of that, and it's the assay and study of DNA and RNA through informatics tools, and as well as starting with genomic assays and stuff like that. But we're predominantly concerned with LC-MS-based assays. Proteomics, we would be studying proteins, and mostly when people are talking about proteomics, we're talking about the study of proteins by proxy of the analysis of their peptides. And then metabolomics, we are generally speaking about looking at small molecules or metabolites. Originally it was coined metabolites, but metabolomics was coined metabolomics for the study of metabolites. But it's generally understood now that metabolomics means the assay of small molecules and metabolites, and oftentimes people group the study of lipids into this category. Here at Masonic Cancer Center, our direct interest has predominantly been in the area of DNA adductomics, or covalently modified DNA. So the main focus for my work and the work of the people that I've been interacting with has been through liquid chromatography, high resolution mass spectrometry, we call it LC-MS for short. And then predominantly in the beginning, we were looking at DNA adductomics, and then using this technical platform. And recently we've been branching out in performing more and more metabolomics or exposomics experiments. So the exposome can be defined kind of as the complex exposure to a multitude of different compounds that we might receive in our body or organism, and they can affect our genome and our metabolism. They come from a variety of different backgrounds, hormones, lipids, anxiety. What we have been interested in, or what I've been working with Rob Tereski and his group with, are predominantly surrounding cooked meat carcinogens, such as FIP and 4-aminobiphenyl, and then also those carcinogens, renal carcinogens, and derived from herbal medicine, such as the ristolochic acid. So if we were to define metabolomics and DNA adductomics as we put to practice in the laboratory, and as I put to practice in my analysis of data, I describe metabolomics as the unbiased and untargeted simultaneous detection of many small molecules. We use LC-MS to screen for known and unknown small molecules, and I say LC-MS squared, and I'll describe that in a second. DNA adductomics is the unbiased and targeted simultaneous detection of many modified DNA nucleosides. Uses LC-MS to screen for known and unknown DNA adducts. The LC-MS technology works like this. We have a sample. However we extract our molecules from our sample, there are many different methods depending on the class of molecules we're trying to study. But after these sample extractions, we have our sample, which contain many, hopefully, most likely thousands of molecules in them. They are then resolved on a liquid chromatography platform and then directly injected from that into a mass spectrometer over time, and the eluting peaks over time can then be selected for mass spectrometry and identification. Well, fragmentation, and then we use the fragmentation to perform identification. And so it works something like this. The molecules are eluting off over time. A molecule at a certain point of time is selected and produces several molecules are looked at the same time in terms of their MS1 level. But a molecule is then selected based on its mass from that initial MS1 spectrum. It is fragmented so that we can later go back and help to identify what that particular compound is. And sometimes we actually take the resultant fragment ions, which might represent a moiety of a compound that has been knocked off under fragmentation, and we further fragment it. And some of these techniques, as has been demonstrated in the community, can actually help us to more accurately identify the compound that we are dealing with. But the challenge in DNA adductomics is not a lot of these compounds are present as DNA adducts in a library, and so we don't have fragmentation spectrum for them, and that's one of the subjects that we'll be talking about today. So DNA adduct detection by LCMS, just to branch off a little bit, it's a little bit more complex than at the level of the MS1 and MS2 spectrum. So here we're generally speaking about broad-based usage of the technology, such as in metabolomics. But things are a little bit different for DNA adduct detection by LCMS. We start with DNA extraction, we hydrolyze them to individual modified nucleosides, enrichment steps take place, and then we inject them into the mass spectrometer. But what's most important is the MS1 and MS2 level data. In most cases, when we're dealing with DNA adducts and I'm trying to help identify them in our data, we are first looking at a very labile, easy to knock off with low energy deoxyribose, which ends up producing a characteristic shift or neutral loss of 116 Daltons, where we go from our precursor ion to our nucleoside without the nucleotide without the ribose on it. So we can use that information in an algorithm, for example, to help us mine for and look for DNA adducts and data. So with that basic knowledge, or that base of knowledge, I can demonstrate to you a couple of projects that we've used custom integrative workflows that we developed to complete a couple of tasks that were important to our groups here at the Masonic Cancer Center. One of them is the development of a DNA adduct search engine that we're calling it. The other is a recently funded development of a DNA adduct spectra library, so we can actually produce tandem mass spectra and tertiary mass spectra to incorporate into a library and give back to the community, and then also therefore be able to aid identification in future studies. So the DNA adduct search engine that we developed, this was developed per the needs of Rob Tereski's group. We call it WSIM-City. And the reason we developed this is there have been mass spectrometry methods that have been developed for improving sensitivity for detection of DNA adducts. One such method is called wide selected ion monitoring data independent acquisition. And all we really need to know in the context of the needs for informatics is when we kind of alter what is normally done what has normally been done at an instrument in terms of how we tell it to collect data, how to screen molecules and save the data. Oftentimes software that is pre-existing will not work with those new methodologies. And so that was one of the reasons we developed this algorithm. There are other reasons as well. If we were to compare the scanning technology that was developed to what has been out in the community previously, we have a couple of scanning technologies known as dynamic data acquisition, or data dependent acquisition, and data independent acquisition. These have been commonplace in the fields of proteomics and metabolomics for a long time, a couple decades. If we were talking about data dependent acquisition, we are typically talking about at a given point in time during our chromatography run, as we elute into the mass spectrometer, we take a full range scan of the masses coming out to the coming into the mass spectrometer, and then we select a few peaks based on their abundances typically. to Fragment so that we can help identify what the peptides in our small molecules are. That was the de facto standard in scanning technologies for quite some time. And then sometime about 10, 14 years ago, data independent acquisition methodologies were developed, and this was to help better improve the coverage of peptides and small molecules that could be obtained in a single scan. So you can imagine here, there's a bunch of masses in this particular stick right there, and we're only selecting a few of them for MSMS. And this one, it's all in, and the only limiting factor is the computational methods at the time to identify what the peptides and small molecules were, and then also lower abundant molecules. We just couldn't really identify them anyway, but you're getting all that information in a single run. Well, what was developed by Rob's group and Pete and James Shubguel was an adaptation of WSIM technology to DNA adductomics. And so how WSIM DIA works is, instead of taking a full scan, we're taking 30 or 20 M/Z zoom ranges to gain some, or to select some, just a smaller amount of ions, but we're boosting the sensitivities for those ions. And then we're performing this DIA-based MS2 evaluation of what we select. WSIM technologies have been around for a while. The method has existed and exists in different flavors. It's pretty interesting to see. I believe the first usage of the physical technology, and it was called gas phase fractionation. That's really what we're calling it today. Has existed for the use of proteomics for a while. So a few people have used it and never really gained hold until really recently, right around this time here. It has been used in the area of metabolomics, but again, not really many people have used it. But what people are starting to see is that it's very valuable for its ability to increase the number of compounds that we can identify in a sample, because we are boosting the sensitivity for those compounds or those peptides in that sample. The WSIM application, WSIM DIA, as applied by Tresky's group, is the first application of this in DNA adductomic work. And it's really, really important for DNA adduct detection. And this is really starts to get into why we need more novel techniques and an improvement in existing tools and techniques to help improve our detection of DNA adducts. Well, DNA adducts are very low level when we compare them to what we are typically have been looking at in terms of abundance in our samples. And if we were to see how low they are in a typical mass spectrometry experiment, if we're looking at this MS1 level scan, this is a precursor monoisotopic peak for a deoxyguanine C8,4 aminobiphenyl in its first primary isotope. And this is considered, it can be considered kind of a high abundant molecule for a DNA adduct in a mass spectrometry experiment. But really what we're starting to see is we're having to deal with being able to detect these peaks in more noisy data, and also down towards the level of noise. And while this monoisotope, we can certainly use only monoisotopic peaks, it helps to also identify isotopic peaks, because having both help us to more accurately identify what compounds might be through formula generation or generation of the atomic formula of those compounds, because we can leverage the ratio between the first two isotopic peaks. But this isotope here, as you can see, is approaching the levels of noise in mass spectrometry, and that's very challenging for common software tools to be able to pick out these peaks without introducing a lot of false positive peaks in our data analysis. This here is just a scan of the MS2 level. Our MS2 level data tends to typically always be much more clean than our MS1 level information, but we need both in order to accurately make an assessment that we are observing a DNA adduct or not. So, typical software was incompatible with this scanning technology. We're getting a sense with the increased interest in that scanning technology by Thermo Fisher, for example, and some other users in the community for metabolomics and proteomics, that the vendors are likely heeding this issue and are making their software more planning to make their software adaptable with this type of scanning technology. Low abundant molecules were difficult to detect using typical automated tools. If we were trying to adapt existing software for the detection of DNA adducts, lower abundant molecules still were very, very difficult to detect. We also needed some sort of sorting and scoring system of results. So if we're mining all this data with an improved workflow, what we hope is an improved workflow, we need a way to really evaluate what the putative unknown DNA adducts that we hope to identify using the software are. So we developed a software to do all of this, and analyzed a number of different data sets. There's a couple of human data sets, human samples that we looked at. There was a test data set where we took 20, Rob's group took 20 spike-in DNA adducts and put them into calf-thymus DNA. And so they serve as a good evaluative measure of the success of our software. And then also putting the software to work more recently is now we're taking data that is being produced in the arena of a study, smoking, non-smoking study, and also dietary intake of rislactins. So the way the algorithm works is we won't go into too much detail about this. The big picture that I really just want to give you is that oftentimes when we're tasked with developing an informatic workflow, we have to deal first with coming up with a scheme to handle our data and how we're going to first hopefully leverage existing software to our best ability, which we originally did, but we have since made some changes. We come up with a scheme and we perform a series of steps, and then hopefully at the these series of steps actually take advantage of known events that happen when we're dealing with DNA adducts. We do something called feature finding, where let's say all of these peaks here, they're the same mass, but the software has to say, hey, they all belong to the same molecule or molecular species or ionic species, so on and so forth. Then we have to somehow aggregate all this data and say who are the isotopes that belong to each other, and then who are the adducts, DNA aglycone peaks, or the peaks after we've lost our deoxyribose belong to the precursors in our WSIM MS1 data. We have to have a way to score these so we can better facilitate the end user going through hopefully what is not thousands and thousands of false positives, but hopefully just a handful of true positives and a handful of false positives. And then perform a series of informatics steps to aid filtering and coming up with a short list of adducts. In the process of the workflow, this is kind of what's going on behind the scenes, and commonly you'll see things like this in other applications as well, but you'll never really see these details. But what the end users end up seeing are things like DNA adductome maps. And so, for example, when we put our search engine to practice and analyzed a kidney sample, we can identify the landscape of DNA adducts that are in a particular sample. Putative DNA adducts are in a particular sample. Many of them are unknown. That's these clear circles here. Some are definitely false positives. We have ways to deal with that. And some are already previously known DNA adducts. And so we can use software to help tell us if we're seeing the real known adducts previously, and then use other output to help us determine which are the best candidates to pursue for identification. So when we actually analyze something like calf thymus DNA that has had DNA adducts spiked into it at various levels, we can, what we're looking for is detection across a broad range of spike-in levels, which would indicate our ability to detect DNA adducts in real samples at potentially very low levels. And so here these adducts that were spiked in range from 0.4 all the way up to 8 adducts per 10 to the 8th nucleotides. So one true test is whether or not, if we were to ask, what can we see if we manually go in and look at our data, and what can be found by WSIM-City in our algorithm? We actually have a fairly high success rate for identification of these DNA adducts. And so in this case, this is that same ctDNA that was spiked in with 3 different levels of various adducts. Some were labeled and some were not labeled. The labeled ones have a different mass loss. If we tell the algorithm to look for the different mass loss, we could identify those compounds also. Compounds come from a variety of classes, and first thing you'll notice is that if we're looking at the manually extracted intensities of the compounds, they do what they're supposed to do. They decrease across the different levels of spike-ins, but every once in a while, if we try to manually identify something, we just can't see it. And so if we're missing out with the algorithm, that's not necessarily a false positive, because for whatever reason, it might not, this aglycone peak may not actually be seen to begin with. And so when we factor in all of these issues, we can see that our overall manual extraction true positive rate, given these issues, can hover right around 80 to 90%, depending on the number of isotopic peaks that we are detecting for the precursor and the aglycone peak. Again, to redefine the aglycone peak, when I say aglycone, I'm talking about a DNA adduct that has undergone fragmentation at a low enough energy to knock off that deoxyribose. So this number is sort of in the background. This number, because we've changed the software, we've improved how well it detects peaks to begin with, and how well it tends to sense DNA adducts from these types of studies. The number is hovering around 90% success rate. It goes down a little bit if we're dealing with lower abundant molecules at the low level, and it goes up a little bit if we're dealing with the more higher abundant molecules. One thing we found out in this entire study is that the existing feature finding tools, the software that actually does this aggregation, was a limiting factor. So feature finding, as I defined earlier, is the step where multiple peaks are assigned to a single molecule. Previously, we were using external software, because you always want to try what exists to begin with to perform this step or any other step. So what we found is we had to take that out of the loop, and we're still detailing the final performance measures of that, but it's definitely increasing our ability to detect very low abundant molecules. So if we put that latest version to practice, we can look at some more recent data that was produced and look at the total number of spike-ins as a true positive measure that were recovered and confirmed by visual inspection, and then also identified by the algorithm itself. We can get a much more much more current snapshot given the software's latest version on its ability to pick low abundant molecules out of both ctDNA and then also out of real human samples. Really the performance that we're looking for is how well can we identify adducts in human tissue. And that's extremely important because that's the reality of what we're trying to develop in the first place. So our false positive rate, if we were to ask how well an algorithm is detecting DNA adducts in a sample, we have to actually visually inspect peaks that are being paired with, or excuse me, peaks that are being identified as DNA adducts with our software. We have to manually confirm MS1 and MS2 spectra as being true or false. We have a number of scoring metrics which help us kind of boost or not boost, but put the real stuff up towards the top of our list. And then also manual inspection helps us to find these false hits, and they tend to score a little bit more poorly. So if we were to look at a more recent ctDNA study and then also apply our software to some human samples after filtering and application of our software, we're identifying a number of peaks that are upwards in the range of putative DNA adducts that include the spike-ins here. They're upwards of 100 plus in the ctDNA study. Some of those might include the actual isotopes being selected as separate DNA adducts. So that's something we have to deal with. If we deal with real data, real human sample, we had a sample acquired from a study and from two different fractions, the lipoperoxide and bulky aromatic amine fractions. We were identifying roughly 40 plus to 60 plus putative DNA adducts in these samples. When we go in and look really at what is being produced and we think are false positives, we actually go through every single one of these hits and we tabulate by visual inspection, and we find that, and this is where I'm really happy, is that the false positive rate appears to be roughly around 20%. That might seem high, but if we were using kind of other software feature finding in our workflow, these lists would be counts would be in the hundreds, so several hundred to 800 or 900 depending on the sample. So now we have much more realistic, much more manageable counts or putative adducts of which we can pursue. In our false positive rates, they might appear high, but they're consistent. And when we're dealing 20% of 100 or 20% of 40, that's not a lot to ask for to sit down over a couple of hours and a cup of coffee and say, aha, we have a list of compounds that we think are real. So previously, we've been able to demonstrate that we can detect known adducts in the software, in prostate, bladder, and kidney. So these are just example outputs of our plots for those particular compounds that were seen in those human tissues. What you're looking at when you typically see something like this is we have a the red or the blue color would be the precursor peak, and then the red color would be the aglycone peak. And so these are good visual tools for confirmation of hey, we have a DNA addict. We happen to know these are definitely the correct signatures because we spiked in a labeled addict for these particular addicts, then they elute at the same time. So when we put to practice the algorithm, there's it's much more complex than this, but I just wanted to briefly share with you some results that have been recently produced from a kidney study. And what we found is using the workflow and using what we know about the false positive rate and performance of the algorithm, we can then assemble quantitative profiles using the algorithm of DNA addicts across multiple samples, and then look for trends and changes of those DNA addicts. And so in this case, we've only selected a DNA addicts that we were able to identify across 6 samples in this pilot study for statistical purposes. And then also we only accepted those that were found across all 6 samples. So we have 3 non-smokers and 3 smokers. In order for machine language and pattern finding tools to work, we have to make sure that we have 100% data present. And so there's a lot more that could go into an analysis like this. But if we just choose this core number of addicts, we end up with a list, 100 long or so, of which we can then do machine learning. And we can definitely see separation between a smoking and non-smoking group. The tool, however, which is really convenient, allows us to readily identify knowns that might be in the sample. We don't know for sure until we actually either perform tandem mass spectrometry and fragmentation and then compare it to a known standard, or spike in a known standard and compare it to its solution time and its mass in this particular sample. So there are multiple ways to better confirm what we are observing, but if we just say based on mass alone, we're likely observing potentially a number of known addicts in this type of fraction. In this process, we're very happy to say that we're starting to see what are realistic peaks in the realistic DNA addicts, putative DNA addicts found in tissues like kidney. And so, for example, here are 3 markers or DNA addicts that might be drivers of differentiation between the 2 groups. Now, we take that as a grain of salt because as we expand out and identify more samples, these statistics might change, but the point is that we can use the underlying software tool to develop an underlying addict profile for a given sample, and then we can extract the quantitative information and look for differences between groups of samples. So WSIM-City is an algorithm to support that provides support for untargeted DNA addict discovery. It now has built-in feature finding. We've made improvements to lower the false positive rate for addict detection. It helps to streamline the data processing, and it provides more efficient usage of hardware. Now, when we're talking about developing an informatics workflow or a algorithm, when I first developed the first iteration of this software, I developed it in a way that was looking, it was a little less efficient. And so what I found is when I started to work on real human data, it would inundate the computer hardware with memory, using all the memory, and crashed my computer several times. So I had to come up with ways to make it more efficient. In our more latest iteration of the software, which we're assaying human samples with now, definitely puts to practice a more efficient usage of the hardware. Well, you might say, well, why not just put it on a cluster and have the big supercomputers do the work for us if that's the case. One goal I typically have when I develop software for researchers is I want it to be usable by those researchers with a minimal amount of resources necessary. So that requires two things. It requires the software to be user-friendly. Oftentimes the software is not user-friendly. And it also has to be programmed well and better able to use a minimum of resources or computer resources. So we're working on the usability by non-informaticists component of it, but now we've pretty much finalized our efficiencies in the development of the software. So another showcase that we wanted to talk about was the DNA adduct library. As I mentioned earlier, there are no shared public resources to get tandem mass spectra to help identify what we might be seeing in our samples as DNA adducts. Well, recently we've been funded a R03. Jingshu Guo is the PI for this particular project, and it's called the development of a DNA adductome database. This is very exciting, I think, because it's something that multiple collaborators around the world have seen as necessary and are contributing by sourcing and sending their DNA adducts that they're known for in their work to our labs, both here and then also at Florida International University, who uses a slightly different mass spectrometry platform. So we can get complementary results off of different technology platforms. If you look at the introduction or the introduction to the proposal, what we're doing is essentially, the reason is DNA adductomics is an emerging field in human biomonitoring and risk assessment, but the absence of publicly available bases is limiting the power of the technology. And so this is the first foothold. The results of this, this work by Jingshu et al. Is establishing that foothold to making a DNA adductomic database. So we're doing this with help from, so when we wrote the application, we gained letters of support from some pretty well-established folks in the area of metabolomics. So to see that the people from that discipline are excited as well, it is very encouraging. So we are getting help from David Wishart from the Human Metabolome Database. He has said he'll help make sure that we can get our adducts visible on his database. Mass Bank, Oliver Feen, this is the primary point of contact for our ability to be able to put our spectra into a public repository. And Oliver Feen is the PI and director in charge of the Mass Bank of North America. So we will be submitting our spectra to him and his group. We also have committed help from Thermo Finnegan to help put this into MZ Cloud, which is just another identification tool that people use when they're doing mass spectrometry. And we're definitely putting everything that we observe into PubChem as what they call a PubChem substance. And if that substance has never been seen in PubChem, it gets elevated to a novel compound. And so with really what is a minimum amount of work on the informatics side, we have to, process the spectra and submit it to mass bank, and then submit information to PubChem, and give data to Thermo Finnegan. We can actually have this data propagate out to a multitude of other different repositories online that actually draw their information from these vital sources. We are working closely with this, for this database with Marcus Cook and Anthony DiCaprio at Florida International University. The idea is that we want to gain additional insight as to how compounds fragment, because on different mass spectrometry technologies, compounds can fragment a little bit differently. And so having that complementary technology allows us to target two different audiences. We predominantly use what we ion trap mass spectrometry. Another pool of scientists out there use predominantly quadrupole time of flight mass spectrometry. So aim one is to collect adducts and data at the University of Minnesota and FIU, and this has been actively underway for several months now. Aim two, in terms of how do we get informatics to help, is to develop an automated workflow to process the MS2 and 3 data, to curate the spectra, and to submit that library to a repository. I won't go into too much details, but it was actually not too hard to develop. We were able to develop an automated workflow. It's kind of working out the kinks right now, but essentially we have a data preparation workflow, which gives us some diagnostic plots, then writes each of the spectra out into a format that another tool by Oliver Fien's group called MS Finder and Riken, in Japan. They've produced a software which will actually help annotate fragment ions and assign structures to them. Well, we have multiple spectra that are produced for every collision type and for different collision energies. And I know I didn't go into too much detail about that, but if, let's say, we have 1,000 spectra, there might be 5 different energy and collision type combinations, and we have to get the best of them and build a consensus spectra, and then have only those 5 spectra present as our final spectrum, a series of spectra for that compound. And then we have to be able to write a report or generate a report so that the user can understand what they're looking at. So consensus spectrum is the concept where if we have a compound and we've produced a spectrum, one typically is not good enough. It has been shown that if you collect multiple spectra for that one compound under the same conditions in the mass spectrometer, the noise peaks can get filtered out, and you can settle on a mean fragment ion intensity that can actually help you score better or produce better search results when you're actually using a spectra and then trying to identify compounds in your samples with this library entry. So the idea is that we can improve the quality of the match by filtering out kind of noise, infrequent noise, and averaging out highly reproducible fragment ions that are produced in the mass spectrometer for a DNA adduct. So you can build libraries with a single spectrum, but the problem with that is you run the risk of it having noise peaks and contaminant ions in your actual spectrum as well. Automated qualitative analysis is part of our output for this particular workflow. One is a total ion current plot. So what we're looking for is when Pete and Jingshu run these standards that we have a profile that increases and then decreases. That indicates the actual standard has been injected into the mass spectrometer, and that they are of sufficient abundance. We're looking for a particular compound, we know its exact mass. We're looking for a performance in mass accuracy that is unwavering across the area where the compound was injected. We have ways to automatically assess the purity. So if one of the first things we do is for a given compound, we assess how much noise in the area or mass range that was used to select our compound, how much noise is persistent. And if there's what appears to be a large level of noise by number, we also look at what is the relative height of the next highest peak. And so in this case, our adduct is about 88% pure, but the next highest peak is only 1% or less of the total DNA adduct peak. And so that's a very good result. Consensus spectrum building looks something like this. For a collision type and for a collision energy, at lower collision types and lower energies, we tend to see our precursor in our A glycone. The consensus spectrum builder produces a formula and a structure, and it annotates those peaks for us. It filters out lots of noise. And then also we kind of track the variance and how much variance we can produce for fragment ion, and this sort of range, these sorts of ranges can be expected in mass spectrometry. If we go to a higher energy, in this case, we expect to see much more complex, much more complex spectra depending on the structure of the molecule, and lower masses, because we're now breaking this down into much smaller components because of the high energy. So we've been able to develop automated processing of direct injection MS data. We've we've provided built-in quality assurance checks in our workflow. We do plot generation, and we can do fragment ion identification. Molecular formula is based on the molecular formula at this moment, but we do have structures that are assigned to each of our fragment ions. We can build a consensus spectra to better improve the quality of our output spectra, and then we automatically generate reports. And so we're still tweaking out the details of how well this will work. So the idea is that when we're done, we will take this information and upload it into the public repository, and it will be available to the general audience. Just to give you a little hint of some other things that we propose to do with the informatics at the ABSR is there has been an increased interest in doing metabolomic studies, mostly to see how let's say cells respond to a dose with a genotoxicant like N-hydroxy-FIP, for example. Typically, what is done in metabolomics in terms of software, however, is that we have vendor-based software, and then we have freeware. And oftentimes, what I have found is there can be shortfalls in any software, be it vendor or otherwise. The vendor software will do multiple steps for us. It'll help us get through data quality assurance more quickly. It'll help us find what the compounds are. It'll help us put names to those compounds, and it'll somewhat help us to be able to do pathways analysis. Oftentimes we struggle with certain steps in vendor-based software, and so we have to do some rescue steps using more custom algorithmic-based approaches that are rather simple to apply. And then the result of that is we can then export data regardless of whether we have to do some data rescue and do some more thorough investigative statistical analysis using other tools that are available to us. And so these sorts of analysis steps are, it may seem daunting at first, but we have the expertise and know-how of how to get rapidly really from here out to something like a software package like this. So just a really, really quick rundown. Here's a study where that was done in the lab for an ASMS poster last year in Rob's group. Two people I mostly worked with were Jingshu Guo and Majda Bahamri. They were studying the oxidative stress-related metabolome changes by N-hydroxy-Fib. And we were able to kind of help them get through some of the data analysis, and then also produce some plots and some statistics and some meaningful pathways analysis. These are things that might seem daunting, like I said earlier, but they're really not that hard to do, but sometimes we just need a little guidance to get going in the right direction. So proposed services, I showed you several kind of what we've done and what our capabilities are on a very complex nature, a medium complexity nature in terms of the database building, and then a more simple nature in terms of just adopting common software results and improving on them and doing some statistics. We have a number of proposed services that we are thinking about. What really comes to mind is support for applications for funding. So sometimes you might need incorporation of principal informatics and data analysis workflows specific to mass spectrometry. In our recent past, we've been able to produce a successful funded application from some informatics aims that were introduced. Of course, that just speaks only to the fact that there's good science on the other aims, of course, too. But what we're starting to see is a lot of people going into the omics realm definitely are having informatics as a component of their work. We can talk about more specific algorithm and workflow development specific to area needs, to your specific areas of research. And then, of course, on a more short-term and minor level, data analysis and statistical support. And we're trying to keep this related to mass spectrometry data sets and LCMS-based omics projects. So in conclusion, computational resources for LCMS data analysis and custom informatics are increasing in demand. Our capabilities at informatics at ABSR under Pete include development of computational resources and workflows, data analysis, grant writing, and consultation. With that, the acknowledgements to the many great folks who are contributing to my well-being, and I thank them very much for giving me the opportunity to work with them. That's it.
Thanks, Scott.
Yeah.
If there are any questions, we can have you type them in the Q&A module and we'll read them aloud. Otherwise, you could raise your hand in the raise hand feature and we can unmute you. Let us know what you think if you have any questions. Okay, we got a raised hand here. I'm going to allow RP Dator to talk. You can unmute yourself now and speak.
Hi, Scott.
Hi.
Morning. So I'm just wondering, how would you, search your WSIM-City mass spectral data that you obtain from your LCMS workflow to your database?
Oh, how would I search it to the database?
Yes, because your WSIM-City could be probably, there's a lot of, you're fragmenting it in a wide range. So how would you know which are the fragment times coming from?
Yeah.
Precursor ion?
Yeah, I mean, so there's a lot of complex issues that can happen in the instrument to begin with. So, in-source fragmentation and stuff like that. The best way to incorporate something like this at the moment is to generate the candidates, the putative candidates, and then to go back in a follow-up experiment and to further evaluate those candidates with higher energy levels at MS2. And so it's those higher energy levels at MS2 that we're likely to garner specificity and matches against a library entry, right?
Okay.
Yeah.
And next question. Sorry.
Yeah. Yeah.
How would you address those adducts that are produced by, like depurination or without the sugar?
So this particular software was developed specifically to look at a characteristic neutral loss.
Okay.
So one thing that could be happening is if there's a specific loss that occurs in a class of compounds that do not have the deoxyribose, and it occurs at a specific energy level, you could possibly do the same experiment catered to those class of molecules, and then we would, in a biased fashion, have to tell the algorithm to search for those losses of different masses. So when I say we're looking, for a deoxyribose loss, we can actually enter any mass we want.
Okay.
Or addition. We can look for associated adducts if we wanted, or we could actually use it to look for within MS1 scans, for example, to look for isotopes.
Okay. Thank you.
Yep, you're welcome.
Any other questions? Got a raised hand from Dr. Volota. 1 second here. And you can talk now, Pete. You might have to unmute on your side. There you go.
Hey, Scott.
Hi.
Just to follow up on what you were saying, I mean, I feel like you could simply just put in the neutral loss of the base of interest, and one could probably directly analyze the same set of data. I mean, the collision energies or type may not be optimal, but so that's just one thought. And the other thing is, and this is kind of related, that this approach, though, the WSIM MS2 and your workflow, data processing workflow, I feel like could be adjusted to kind of account, to look for any sort of neutral loss that anyone in the cancer center is interested in, such as, I mean, the simplest example would be RNA adducts, or you could do things like anything like derivatizing classes of compounds and looking for the derivatizing agent neutral loss, or maybe classes of metabolites, etc. So any sort of screening of a class of compounds that have a constant neutral loss. So could you speak to that a little bit, maybe, or what do you think of that?
Oh yeah, no, so the software as it exists right now, it definitely can be used on any data set. You can just plug in any other neutral loss that you're interested in. You can also, retroactively go back to any of these data sets that have been produced using this technology. If we're talking about constant neutral loss DDA, it's a little bit different. I haven't looked too much into that. So we're talking about a different scanning technology now. I think you were alluding to that perhaps a little bit. But if others are thinking of that as well, one of the things that we leverage to reduce our false positives is at the MS2 level, we need to have an eluting peak so that we can get a much higher scoring true positive to better separate it out from the false positives. But yes, we can alter the parameters of the algorithm to look for basically any neutral loss within the same sample.
All right. Well, thanks to Dr. Walmsley for giving his presentation, and thanks to everyone for joining us today via Zoom. This concludes our program. Thank you so much. Have a good day.